So today I'm going to first talk a little, give a small introduction to Lanza and into nucleofection and CAR-T. Then I'm going to go into nucleofection in detail to show you how it differs from the other transfection technologies in some fundamental and mechanistic sort of ways, so that you can see how, one, how it's different from these other technologies, and then how we can use these differences to help transfect clinically relevant cells and in clinically relevant numbers with these genome editing substrates and other things. Then we'll dive into nucleofection devices and systems. Briefly, I'll talk about the 4D nucleofector and our new LV system, our large volume transfection system, which allows you to transfect clinically relevant numbers of cells. So this is the latest addition to our nucleofection platform, and one that really fits very nicely into the cell therapy field. And so with this introduction, we can then look at CRISPR and nucleofection in detail to see what people have been doing with nucleofection and CRISPR and how they've been able to really change things and maximize the editing efficiencies and things like that. But first we'll start with a little introduction to Lanza. Lanza is known for primary cells. That is one of our biggest parts of our portfolio. And we have our clonetics cells, which are terminally differentiated cells from every part of the body. Not only the cells, but also the medias in which to grow these cells. We're very good at media formulation, but it's important with primary cells to have the right media. They have special needs, special growth factors and cytokines that they need. So 10% serum and DMEM is not necessarily going to be sufficient for these primary cells. But with Lanza, we have formulated medias where these primary cells are guaranteed to grow and thrive and survive. And so this makes working with primary cells very easy. You can get our cells, you can get our media, and they're guaranteed to work together very nicely. Our Poetics line is our adult stem cells, and our immune system cells also fall into this category. Again, we have medias in which to grow the cells, but in many cases we also have differentiation medias where we can differentiate things down the different pathways. We have disease state cells for respiratory diseases like asthma, COPD, and cystic fibrosis, as well as diabetes, type 1 and type 2. And we also have a custom isolation service. So if there's a cell type you would like to work with, but you don't see it in our catalog, talk to our representatives and we can work with you to see if, put you in touch with our cells on demand people, and see if we can find these cells and isolate them for you, how much this would cost, and price this out with certain milestones. So when it comes to cells, there's a lot that Lanza has to offer. In 2008, Lanza acquired Amaxa, legacy Amaxa, and they for the nucleofector technology, because nucleofection excels at the transfection of primary cells. So it was a very synergistic acquisition. Lanza has primary cells, Amaxa has the ability to transfect these cells, and so it was a very good acquisition. And so these two fields interact very closely with each other. We also have a variety of other systems and downstream assays. We have our RAF3D cell culture media, which is important when we talk about next generation cell-based assays, and I'll touch on that briefly in my talk. We also have MycoAlert, mycoplasma detection system, which is very important because mycoplasma is a major contaminant that one must be aware of, and a variety of other downstream assays. But let's focus primarily on where Lanza can fit in the immunotherapy field. So Lanza has hematopoietic and immune system cells that are available for preclinical research, and as you build up towards your therapies, you can use these cells to help define things. Nucleofector technology allows you to manipulate and transfect these cells. And then we have downstream accessories, like I mentioned, the RAF3D cell culture. We have CytoSmart, which is an incubator monitoring system. But we also have ex vivo media, which is available both in research grade as well as GMP grade. And this is an ideal media for the expansion of T cells. This is an important part of any immunotherapy. After you do your modifications to expand your cells, and ex vivo is an excellent media for that, serum-free, defined media that is very useful in this context. So when we started looking at Lanza, We can help out in so many different ways. We have a tremendous amount of expertise and many different divisions that all interact to assist you as you move from the research into preclinical and then clinical and finally commercial modes. So we have our primary cells that I've been talking about. So this would be ideal for research and into the preclinical fields where you need cells that are consistent. You can work on this and derive your therapies. Our sister division, Emerging Technologies, has GMP grade cells that are available, so hematopoietic and immune system cells that would also be of use. We have our transfection technology, of course, the 4D nucleofector X unit and our LV nucleofector. So the 4D would be primarily for research and into the preclinical realm. The LV would pick up in the preclinical realm, but also the clinical use of the LV as a therapy, and then also even into the commercial aspect as well. CytoSmart can monitor cells throughout this entire process. We also have medias. We have serum-free medias that could be used in research and preclinical work, as well as the serum-free medias that are available in a GMP grade from the preclinical on through the clinical and commercial realm. And finally, LONZA is also known for endotoxin testing. This is on the regulated side of the industry and more on the manufacturing side, but basically any parenteral drug, anything that gets injected into a person needs to be tested for the presence of bacterial endotoxins. And LONZA is a leader in this field, and we have this capability as well. So as you move into a cell therapy mode and you create these cells, which are essentially the new drug, these also have to be injected into people and have to be tested for the presence of bacterial endotoxin. And that is, again, something at which we are expert. So there are many different touch points. As you move from the bench into the clinic, there are many touch points where LONZA will be able to help you, and we have a tremendous amount of expertise. We have unique products that can assist you along the way. And when it comes to regulatory issues, we also have a great deal of experience. So LONZA can help in many different ways, not just my particular division, but our other divisions as well. So let's move on to nucleofection. Our 4D nucleofector is an advanced form of electroporation. We've got the nucleofector device. We also have cell type specific nucleofector kits, nucleofector solutions, which again, cell type specific, so we find different solutions will match up with different cells. We have detailed optimized protocols, but essentially what happens with nucleofection is we have excellent transfection efficiency, but high functionality and high cell viability. And as we get into the mechanistic details, you'll see how this plays out. We are also moving in the clinical direction. I have to say at this moment that the LV nucleofector is for research use only. However, many of our customers are also taking the LV unit and moving in the direction of clinical trials with this. They're submitting INDEs, investigational new drug exceptions. They're submitting these to the US FDA. And if these are approved, then this would allow the researchers to use research-grade reagents in their therapies. And that's what we have with the LV nucleofector. Several of our customers have already applied for these INDEs, and we're in the process of working through this situation, and they are looking at clinical trials in 2018. So in support of this, it turns out the more that this is looked at, there's not a lot of guidance out there from the FDA on GMP manufacture of something like the nucleofector device. Is looking more and more like an accessory device, and there's a different regulatory burden on an accessory. And so what we have right now is where we have supporting quality documentation, advanced documentation on our plastic electrode, which is the only part that is in contact with the cells. So leachability analyses, we can look at sterility and presence of endotoxin and a variety of other things involved in the manufacturing of this in support of using this in clinical trials. We have our logware software, which is an optional software that you can load onto the nucleofector device, which is 21 CFR 11 compliant. So this has a number of features, but among them it restricts access to authorized users. It creates an audit trail. It protects the integrity of the data, so the data cannot be changed or deleted. All the sorts of things that the FDA is looking at as you move forward into the clinical realm. We also have initial qualifications, operational qualifications, tests available for this device. The IOPQs, the initial and operational, the performance testing, the PQ, is left to you. That would be your particular assay, and so you'd have to determine what that is. But we could do the initial qualification and the operational qualification test for you. So in essence, we have a device that is currently for research use only. It is being moved in the clinical direction. Many of our customers are using the LV and intend to go into clinical trials with this. And so that is the current status, but it is looking like we're going to be a key player in the immunotherapy field. CAR-T is everywhere in the news. You see headlines everywhere, not only in scientific journals, but in public journals like The Guardian or CBS News. This is an assay or a therapy that is, in my opinion, going to finally deliver on the promise of biotechnology. After all these decades, we have many successes. We've made improvements in medicine, but I think CAR-T therapy is the first truly revolutionary therapy that's going to come from biotechnology, and I think it deserves all the press that it's getting. The disease that's been treated most often with CAR-T thus far is acute lymphoblastic leukemia, ALL. This is a B cancer, B cell cancer in the blood. It affects young children and adults. And chemotherapy and bone marrow transplant usually work very fine in treating this disease, with an 85 to 90% survival at diagnosis. However, if there's a relapse or multiple relapse, the prognosis gets very poor. And that's what we had here with Emily Whitehead, who was the first child who was treated for ALL using CAR-T. And so she was within hours of complete organ failure and likely death. They infused her with the CAR-T cells, and there was an almost immediate turnaround. And very quickly, she was on the road to recovery, and at this point, she's now 5 years cancer-free. So this is the reason we do this, is the ability to really have tremendous impact on people's lives. So getting back to nucleofection on this, why is nucleofection considered such a good choice for this? And basically, nucleofection has been around for a long time and been transfecting human T cells for a long time. Our first kit was for primary human T cells almost 20 years ago. And this paper is an old one, but it's a good one. It's from 2003, but it shows about nucleofection of cytotoxic T lymphocytes. And in particular, when we look at how the T cells are attacking target cells, it's quite relevant to what we're talking about with CAR-T. Briefly, to go through this paper, these researchers were interested in the release of perforin into target cells, which is involved in the killing of them. They were looking at the role of MUC13-4 in this process. So they made a red fusion protein with MUC13-4. They stained the cells green for perforin, and then if you look in the overlay, you can see that both of these proteins are hanging out together. They're not exactly co-localized. They're both present in the cytoplasm. However, this changes markedly when you put the T cell in contact with the target cell. You see this tremendous co-localization of perforin and MUC13-4 in the middle of the immunological synapse between the two cells, and resulting in a targeted release of perforin directly into the target cell. So this is exactly what we see happening in CAR-T therapies with T cells attacking the target cells. So I figured this was very relevant to what we wanted to talk about. But the point here is that nucleofection has been doing this for a long time. We've been a trusted technology for the transfection of human T cells, and now with the LV unit, we are finally in a position where we can now do this kind of effective transfection with clinically relevant cells and clinically relevant numbers of cells. Nucleofection has been in clinical trials before the FDA. Here are several CAR-T publications for CAR-T work that's been done with clinical trials. Two of these involve Lawrence Cooper from MD Anderson down in Houston, Texas. Dr. Cooper's now moved on to Xiopharm, and he's working on CAR-T there. So basically, nucleofection is not a stranger to clinical trials or the FDA. We had a beta tester at Stanford University, Dr. Matt Porteous, who is really looking at sickle cell disease. So not exactly CAR-T therapy, but again, cell-based therapies using nucleofection. So they're working on using Crispr to repair the gene that causes sickle cell disease. They're looking at clinical trials in 2018. They had a very nice paper, a very thorough paper that came out in Nature in last November 2016, where they used CRISPR to repair the faulty gene. They snipped the faulty gene using CRISPR, and then they used adeno-associated virus to introduce the repair template into hematopoietic progenitor cells. They successfully modified these cells, enriched them, and then implanted them in non-skid mice, and these CD34 cells then produced normal red blood cells. So this looks like a very possible cure for sickle cell disease. So they're basically planning to make an initial submission to the FDA in the next few months. That has actually happened. I should probably change this slide. But they've already submitted this INDE, and they hope to treat the first patients in 2018. So things are moving forward in cell-based therapies, not just CAR T. And then we also have MESCAMs, mesenchymal stem cell therapy for Canadian multiple sclerosis patients. And so this is an international study using mesenchymal stem cells to treat multiple sclerosis. So people are using these adult stem cells or human T cells in very innovative ways, but nucleofection can play a role in every one of these cases. And that's because nucleofection basically transfect every cell type that's out there. When I put this slide together, I was basically hitting the main research areas that people were working on: cancers, neurology, immunology, metabolic disorders, cardiology. I selected representative diseases from each of these areas, and then started listing cells that we can transfect, things we already have optimized protocols for, but cell types that people who are working in these areas might be interested in using in their work, and maybe are not using in their work because they can't transfect or manipulate them. So with cancers, I selected breast and prostate cancers, two prominent cancers, and then so we can transfect mammary or prostate epithelial cells very nicely. With neurology, we can transfect any neuron out there, whether central nervous system or peripheral nervous system. And we can do this in adherence or in suspension. Immunology, we can transfect every cell in the immune system, T cells and B cells, monocytes and macrophages, CD34 hematopoietic progenitor cells, or NK cells. And we can do this in both human and in mouse. So you can choose your model system as needed. Cardiology, I'm a vascular biologist by training, so that's how I first got started with nucleofection, transfecting HUVEC and HEMVEC cells, so you can transfect all the vascular cells as well. So nucleofection is superb and can help you no matter what your field is and whatever cell type you're working with. I've left stem cells and iPS generation for the last because that's a bit of a special case scenario. But because we can transfect primary cells so effectively, we can also transfect in the reprogramming plasmids to these cells and then create iPS cells out of all these different cell types. And then once you generate your stem cells, you'll also want to manipulate them or transfect them. And again, nucleofection is superb on that. So again, nucleofection has the capability to help you no matter what field you're actually working in. So why nucleofection out of all the other transfection technologies out there? And essentially, we have high efficiency transfection of primary cells and physiologically relevant cell lines. So now you don't have to compromise. You can use the most relevant cells for your work. And we think that using higher quality cells, normal primary cells, will yield higher quality data and higher quality of hits. And also, it's ideal for the transfection of all the genome editing technologies like zinc finger talons or CRISPRs. We have tremendous substrate versatility. We really don't care what that substrate is during the transfection. So we can transfect plasmid DNAs like cDNA expression plasmids, siRNAs, DNA oligos, mRNAs, reprogramming vectors, peptides, proteins, morpholinos, Q dots, small molecules. These are all things that people have transfected using nucleofection. We have flexibility of scale. We have our benchtop device, our 4D nucleofector, but you can go from single cuvette up to 96 well or 384 well transfections without having to change technology. You use the same solution and the same program and get the same results. And if you're using the same size transfections as you move across the platform, even the same cell numbers. So it's very versatile. And now with the LV, we can also go up in cell numbers. So this was all made possible because of an innovation in transfection. We moved away from the aluminum electrodes that everyone's using to the a black plastic polymer, conductive polymer electrode. And this allowed us to have a lot of flexibility, allowed us to create a robust 384 well transfection device, adherent transfection in a 24 well capacity for neurons and other cells, and the new LV nucleofector, which allows us to transfect large numbers of cells, from 10 million up to 2 billion cells, and actually for small cells like T cells, all the way up to 4 billion. So the principle of nucleofection is the efficient non-viral transport of DNA straight into the nucleus of primary cells and cell lines. They have a very high gene transfer efficiency, and this transfection is independent of cell division. So if you look at the other transfection technologies out there, standard electroporation, lipids, calcium phosphate, it's easy enough getting DNA in the cytoplasm of the cell, but the rate-limiting step is getting the DNA into the nucleus of the cell, where it needs to be in order to be expressed. So during cell division, the nuclear membrane breaks down. In the daughter cells after cell division, the nuclear membrane reforms, and some of the DNA that was in the cytoplasm is now in the nucleus. But there are consequences to this dependence on cell division. One, you're limited to working with actively dividing cells, so you have a very difficult time transfecting neurons and a variety of other cell types. Also, you have to wait 24 to 48 hours to see good gene expression, because you have to wait for the majority of the population of the cells to undergo cell division. Nucleofection works completely differently. We get DNA into the nucleus immediately upon transfection, independent of cell division. So we can transfect things that don't divide, like neurons, or other difficult cell types like suspension cells, which seem to be difficult for many other folks. We also see faster gene expression, typically 12 to 18 hours for peak GFP expression, but if you transfect cells in the morning, you'll see green cells by the time you go home, and maybe a lot of green cells. And you have this versatility, you can transfect all these different substrates in either primary cells or cell lines. So there's two parts to the nucleofector technology. We have cell type specific solutions that interact with the cell membrane, and then when we apply our nuance pulses, we get the DNA directly into the nucleus of the cell. Now what happens with nucleofection is when we hit the cells with a pulse, we open up pores in the cell membrane and also in the nuclear membrane. Then our cell type specific solutions will stabilize these pores and keep them open for around 15 minutes. So then whatever substrate is present will passively diffuse into the cell. It's not driven in by the electrical pulses. It's a simple passive diffusion event. And so there's things that we can do to play around with this. If you were to, for example, double the dose of substrate, you would increase the concentration gradients and tend to drive more of your substrate into the cells. So in a crude way, just by changing the concentration of the substrate, we can regulate or change the copy number of the substrate getting into the cell. So when we start looking at exotic substrates like the Crispr RNP, these sorts of things will help us make some serious differences over there. Here's some proof that we actually get DNA in the nucleus immediately upon transfection. Here we transfected a very difficult primary cell type, normal human dermal fibroblasts of neonatal origin, transfected them with a GFP expression plasmid that's also been labeled with tetramethylrhodamine. So these red dots that you can kind of see here are the actual DNA molecules inside of the cell. 2 hours after transfection, we fix the cells and stain the nuclei with DAPI. In 2 hours, we already see a reasonable amount of GFP expression in this cell. You do the merge diagram, and you see that we have red dots in the cytoplasm, but we also have red dots in the nucleus. Now this was analyzed by confocal microscopy. So we've taken a digital cross-section through the cells. So we know these red dots are in the nucleus and not above or below the nucleus. So even though I've been talking about how we target DNA to the nucleus, that's not exactly accurate. What we really we end up with is a homogeneous distribution of nucleic acid throughout the cell. There's DNA in the cytoplasm, but there's also DNA in the nucleus, where it needs to be expressed. Now, as we go into the genome editing part, we also transfect other substrates like siRNAs or mRNAs. siRNAs and mRNAs need to be in the cytoplasm in order to be functional or translated. Well, they're nucleic acids. We're going to have the same homogeneous distribution throughout the cell with mRNAs and siRNAs. So they'll be in the cytoplasm where they need to be functional or translated, yet the DNA gets in the nucleus where it needs to be expressed. So it's really the best of both worlds. You can see in this slide and in the previous slide just how nucleofection works and how it's fundamentally different from the other transfection technologies that are out there. So let's move on to nucleofection devices and systems here. We have a platform, like I said, and the LV is the final part of this platform. We have our benchtop device, our 4D nucleofector, which is small scale. If you decide you need larger throughput, higher throughput on this, you have a 96-well shuttle system as well as a 384-well HT Dukla Factor. So you can move on up the scale here without having to change a thing. You use the same solutions, the same programs, and if you're using 20 microliter transfections here, the same cell numbers, all the while getting the same performance. So you can scale things up or scale things down without having to change any of the conditions or re-optimize. If you create an assay with the 4D Dukla Factor, you can feed the cells directly into that same assay using the shuttle or the HT Dukla Factor. So you have apples to apples comparisons as you scale things up or down, and also with the LV. So in this graph, it kind of shows a relationship between throughput and high cell numbers on the different axes here. So we have our 4D Dukla Factor system, and as well as our older Dukla Factor 2B device, which is also still a fine device, which are low throughput and low cell numbers. If you decide you need higher throughput, then you can go with the 96-well shuttle or the HT Dukla Factor for even higher throughput. If you need higher cell numbers, that'll be lower throughput, but still higher cell numbers up on top here. We also have our, I mentioned this before, our logware software, which is 21 CFR 11 compliant. Again, this has all the things that the FDA is looking for as you move in the clinical direction. It restricts access to authorized users, has electronic signatures, it'll generate audit trails and protect the integrity of the data. And this is an optional feature, but this can be loaded onto the 4D Dukla Factor. And so this is very suitable for use of the 4D Dukla Factor in a regulatory environment. So the 4D Dukla Factor, it's basically non-viral transfection, just like the older Dukla Factor 2B. Easy to use, intuitive software, and improved experimental flow compared to the older system, primarily because we have plastic electrodes, which are non-toxic, compared to the aluminum electrodes in the previous version, which leach toxic aluminum ions into the cell suspension. This is something we can, it really improves the experimental flow through because we don't have to worry and rush so much about getting cells out of this toxic environment. This is all a laboratory situation if you have an immunology lab, and some people are working on T cells and B cells or monocytes. You have in the old device, you'd have to buy a different kit for each one of these groups, yet with the 4D, you can use the P3 solution for all of them. Different programs will allow them to transfect their different cell types, but you buy one kit for the entire lab. So it's very much a savings there. And then you can do nucleofection with different cell numbers by using either the strips or the cuvettes. This is the software for the Dukla Factor device. You can see we can either choose strips, which are 2 rows from a 96-well plate, or cuvettes. This uses existing 96-well shuttle protocols. So everything in our database that's been generated with the shuttle or with any device that has the plastic electrode will also work in the 4D. This is particularly useful for work with rare or precious cells, such as iPS cells or human embryonic stem cells. These cells can be very expensive, they grow very slowly, and so they take a long time to accumulate enough numbers for an experiment. The medias are very expensive, so there's a cost as you keep passaging these cells. So if you have to grow up 1 million cells for each transfection with a cuvette, that can be very time-consuming and very expensive. Whereas if you can get your answers with a smaller number of cells, you can transfect 200,000 cells in each well of the strip and get many transfections out of that same 1 million cells. And so now you can operate faster. You don't have to wait so long for a lot of cells to grow up to do your experiments, and you can get by with fewer cells. And so it is really useful for stem cell work. And finally, nucleofection is so efficient at non-viral transfection of primary cells, it's an excellent choice for zinc finger talent, Crispr delivery, or iPS generation as well. Our 4D Dukla Factor, our LV Dukla Factor, is the latest addition to our device. It's part of our modular system. We have the core unit on top, which drives the LV. It's a closed, scalable system and can transfect anywhere from 10 million cells up to 2 billion cells, or even 4 billion small cells like T cells. You can optimize in the 4DX unit and then transfer the conditions exactly over to this device. It can be operated with our 21 CFR 11 compatible logware software, and we've tested this out on a variety of cell types, including human T cells, human CD34 plus cells, and a variety of cell lines. There's two flavors of loading this. We have a 1 mil cartridge that you can load with a P1000, put in 1 mil of your cells and your transfection solution and substrate. You plug this into the device, and then you can activate it and transfect the cells, and then remove them with the P1000. And so we can do up to 100 million cells at a time, or 200 million small cells like T cells. Essentially, you take however many cells you can do in the cuvette, 100 microliter transfection, and multiply it by 10. So with small cells like T cells, we can do 20 million human T cells in our cuvette, which would be 200 million cells in this device, in this cartridge. We also have the flow-through cartridge. It's essentially the same cartridge, but now we have a tubing hooked up to this thing, so that when we plug this into the device, we can run this tubing through two peristaltic pumps. We can feed cells in through the upper pump, and optionally, you can put your substrate in through the lower pump. If you have something that's labile, like mRNAs, you can add that separately so it doesn't have to be mixed in contact with the cells for the duration of the experiment. Then when you program this, it'll feed in 1 mil between these two entry points, feed in 1 mil of the cell suspension into the cartridge, and then transfect it. The air will push this out into a destination syringe or bag or whatever, and then it'll put in the next mil and transfect this. And you can do it 20 times. And so if you can do 200 million cells in one 1 mil cartridge, if you do this 20 times, that would be 4 billion cells in the flow-through cartridge. So you can transfect clinically relevant numbers of cells in a closed system. Nothing gets opened up through here, so it's a complete closed system throughout. As I said before, you can optimize in the X unit. Here we see the modular nature of the device, the core unit on top, the X unit, and then the LV. So you can optimize things in the 4D and then transfer the conditions exactly over to the LV. The same settings regardless of which cartridge you use. So let's start looking at some data here. These are human T cells, and then we tested everything compared to 100 microliter cuvette versus, in this case, the LV unit at 10 mils, and a variety of cell lines as well, 100 microliters to 1 mil in the cartridge, and the same here, or 100 microliters with 20 mils of cells through here. So you compare, we have the same transfection efficiencies and very comparable viabilities when we move from 100 microliters up into 10 mils. And the same thing here with the K562s and the HL60s, and very comparable transfection efficiency and viabilities across the board. Let's take a closer look at human T cells, because that's most relevant to the clinical setting here. You can see we have two programs here, EO115 and FI115, which is a stronger program, which will kill more cells but also have a higher transfection efficiency. EO115 is gentler and will keep the cells more functional. So we compared the 100 microliter cuvette to the 1 mil cartridge to 10 mils of cells through the flow-through cartridge. We see very comparable transfection efficiencies and very comparable viabilities as well. In both cases, higher transfection efficiency with FI115, and aside from a small dip there, very comparable viabilities across the board. But those were all done with GFP. Here's an example of human T cells transfected with the LV using the Crispr RNP. So this was done by Matt Porteous, who was our beta tester for the LV. He's a longtime nucleofection user, and he had worked out all the systems for RNP transfection with his X unit, his 40X unit. So he this is his first comparison. When he got the LV, he decided to immediately leap into the RNP and not worry about controls. So his very first experiment, he transfected in blue the X unit and the LV next to this. Very comparable data, very parallel, especially when you consider that he used twice as much Cas9 with the X unit as opposed to the LV unit. He didn't realize how much Cas9 he needed and didn't have enough on hand, so he just cut the amount in half. So 300 micrograms per mil versus 150, and the results are still practically parallel to each other. He has since duplicated this experiment and then gone on to his CD34 cells, and it worked very nicely. But this was the very first time he tried it, and right out of the gate, he got very nice results using his Crispr RNP. But it's not just CRISPR. This is also from another beta tester using the LV with zinc finger mRNAs. And they transfected actually both GFP mRNAs and then zinc finger mRNAs over here. And then they looked at indel formation using the cell 1 assay and quantified this. And you can see that the X unit got a very nice transfection efficiency. It dropped a little bit with the in the LV unit, but still very respectable indel formation here, editing efficiencies. So both zinc fingers or CRISPRs, all these editing technologies work very well with nucleofection. So now, with that preliminary, let's dive into CRISPR itself. So the genome editing applications with CRISPR, one of the underappreciated applications would be the stable modification of primary cells, such as iPSCs for in vitro disease modeling or cell-based assays. And I really think this is where science is going to be going in the future. I think it's the next generation model systems. So with nucleofection, you can generate iPSCs very effectively. Using CRISPR, you can now introduce a modification in a very specific gene in this. So now you have isogenic population, two isogenic populations of cells, identical except for that one CRISPR modification. These cells can then be differentiated terminally in a variety of different directions. We're getting very good at this, both at Lanza, but in our industry in general. So at Lanza, we've made motor plate neurons and dopaminergic neurons. Some customers of mine have made sensory neurons. We've made hepatocytes in-house and cardiomyocytes. I have a customer who made cardiomyocytes. I have this beautiful movie of this sheet of cardiomyocytes beating synchronously. So even functionally active cells. And we have some customers up in Canada who are differentiating their iPSCs into lung epithelial cells, and then putting these into an air-liquid interface and then watching the further maturation of these cells. So everyone's getting very sophisticated in how they differentiate iPSCs. So then you put this in context with nucleofection and CRISPR. We can modify these iPSCs, and again, isogenic populations, identical except for that CRISPR modification, differentiate these down terminally, and now we have human cells in a dish, identical except for that CRISPR modification, and now we can begin to do science with normal cells. Things that we've only been doing with cell lines in the past, we can now do with normal cells. This is, I think, the model systems for the future. And we can even take this one step further and move these cells now into a 3D architecture like our raft or something else, and really begin to look at these cells in an in vivo-like situation. Those are the model systems of the future, and that's where I think science is going. But we can also do the stable modification of cells like iPSCs or CD34 cells or T cells for clinical applications. And this is cell-based gene therapy, basically autologous replacement and transplantation of disease cells by healthy cells. So you take CD34 cells out of a patient, modify them, and then return them to the patient. So that'd be autologous. The repair replacement of defective genes for sickle cell anemia or beta thalassemia or other well-defined genetic diseases like that. And this is there already are publications out doing this. This is one that came out in Nature in 2014. These folks were using nucleofection to basically cure skid mice. This is the Matt Porteous publication I've mentioned several times already from Nature 2016, where he repaired the beta globin gene in hematopoietic stem cells using nucleofection. Generation of antigen-specific T cells. This is our CAR-T work. And again, putting in chimeric antigen receptors into T cells. When you reinfuse these cells, it'll cause them to target and kill cancer cells very specifically. But also simple things like barcoding cells. So what I mean by barcoding is you can, using CRISPR or one of these other genome editing technologies, introduce a small piece of DNA that you can recognize with PCR primers. So now when you transplant cells into an organism, you can determine definitively whether these cells came from the transplant or if they were from the host that got differentiated in part of the treatment. So this barcoding of cells allows you to mark cells and then definitively show where their origin was. But all this cutting-edge technology can really improve speed to patient, and that's what we're hoping to do with nucleofection and these other aspects of this. Is how fast can we make these sorts of things available to the patients? So looking at nucleofection and gene editing, basically it's a non-viral system. Right now, most of the CAR-T work is being done with viruses. They've had the first start in this, they have a head start, and so they're ahead of the game. But I think anytime you use viruses in a clinical setting, it always raises issues, and you wonder, are these going to cause cancer? What's going to happen with these viruses? So if you can do something in a non-viral fashion, that always seems more attractive to me. As a non-viral technology, it allows easy adaptation to different genome editing systems like zinc finger, TALEN, or CRISPR. We can take multiple substrate types, such as DNA, mRNA, or protein, for these transfections. We have the substrate flexibility and simple co-transfection using the same conditions for all these different substrates. And it's a reliable, proven technology. There's over 80 genome editing publications out, peer-reviewed publications by well-known people in the industry. So it's a very powerful technology. So the possibilities with genome editing are really limited only by your imagination. There's so many different things that can be done. Basically, you can add or delete transcription factor binding sites. You can delete or mutate a promoter or put in a whole new promoter. You can disrupt a gene with a knockout or generate a specific point mutation in an exon. Insert a new exon to make your gene fusions, or fuse a reporter gene to the back end of a gene. Or even add or delete microRNA control elements. So there's so many different ways you can apply this. All these genome editing technologies work in the same fashion. They have a DNA binding domain of some kind that lends specificity and targeting to the complex. And then we have a DNA-specific cleavage molecule that will either nick or cut the DNA, make a double-strand break in the DNA. So if we look at the three main technologies here, we have zinc fingers and talons. They're really very similar to each other. They have different DNA binding motifs, but this is basically protein finding specific DNA sequences. They have the FOC1 nuclease that's fused to this and will nick the DNA. So we need two of these constructs, one on either arm to nick the DNA. So fusion proteins, these are awkward to deal with because there's a lot of algorithms that are needed to figure out exactly which protein sequences we're going to bind which DNA sequences specifically. So companies like Sigma or Transposition have powerful algorithms that they use to figure this out. But this makes it difficult and awkward, and having to make multiple fusion proteins for each change you'd like to make. It's difficult and costly and slow. And this is where CRISPR has really changed everything. Because you have a simple guide RNA that lends specificity, the simple base pair complementarity. Even a summer student or a grad student could design a guide RNA. This then gets complexed with a generic Cas9 protein, which will introduce the double-strand break. So there's no fusion protein needed. It's very fast, very easy, and flexible, and that's why CRISPR is taking over our industry. The reason I mention all this is because nucleofection is a recommended delivery technology for zinc fingers and talons. It has long been this. For example, if you were to call up Sigma tech support and say, I'm having problems with my zinc fingers or with my CRISPR, the first thing they're going to ask you is, are you using nucleofection? Because they use nucleofection for everything. They're easy cells as well as the hard, because it's simple, reproducible, works every time. They don't have to worry if lipids are delivering their construct effectively to this cell line or that cell line. They just use nucleofection for everything because it always works. All the leading CRISPR researchers are also using nucleofection. So Doudna and Charpentier, Zheng Fang, George Church, all these people are using nucleofection for their difficult cell types, stem cells and other things. So we're well situated and well recognized in this field for what we can do. So the basic, whether you're using zinc fingers, talons, or CRISPRs, the mechanism is really the same. A double-strand break is introduced in the DNA, and this is a very dangerous situation for the cells. They have to repair this immediately. And they often do this, well, in order to do this, they induce the DNA repair pathways tremendously, 1,000-fold, some say even 10,000-fold, to induce these pathways to repair this very dangerous situation. And normally they repair this very efficiently, but it is an error-prone process. And sometimes you'll get insertions of a few base pairs or deletions of a few base pairs. These are the indels that people are measuring. And if these can often create a frame shift mutation in your protein, which will then throw off the frame and usually introduce a premature termination of your protein. So this is done by a process called non-homologous end joining, and this induces random mutagenesis here. Alternatively, you can add a piece of template DNA, which will span the double-strand break, and with a different DNA repair pathway, a homology-directed repair will cause the insertion and faithful insertion of the entire piece of DNA, repairing the double-stranded break. So this is what we're looking at with CAR-T, is to use our template DNA in order to repair the break and insert the chimeric antigen receptor. So moving on to nucleofection, basically you're going to need to optimize your substrate ratio of your substrate and the guide RNAs. It's very important to do this, and if you want to maximize the editing efficiency, this will end up becoming critical. We went through all the early publications on nucleofection and the different genome editing technologies to try and get a handle on what people were doing. And you can see with zinc fingers, people in a 20 microliter transfection, most people were using between 0.2 and 1 microgram of DNA for their zinc fingers. Downs was pretty much the same story here, 0.2 to 2 micrograms of plasmid, so pretty much the same ballpark. mRNA, they would use between 0.4 and 4 micrograms of mRNA in a 20 microliter transfection. And then we have a variety of recommendations for donor plasmids and things like that. Moving down to CRISPR, we had people go from as low as 0.5 all the way up to 10 micrograms of DNA in a 20 microliter transfection. Let's talk for a moment about that. I would not recommend using 10 micrograms of DNA in any small transfection like that. DNA, no matter how pure it is, does have DNA toxic effects, DNA-induced toxic effects. And so using this much DNA in a transfection like this would almost certainly cause toxic effects. However, someone did this, they got some data, and they were able to publish it, but I would not recommend that at all. So in Zhenfeng's seminal paper, Ran et al., when he was doing nucleofection of his stem cells, he used 200,000 cells and 1 microgram of total DNA substrate for his CRISPR reactions, and that, I think, is a very good ballpark on where you should be going with this. But people are moving away from DNA transfections and moving more towards transfection of mRNA and RNP with this. And basically, this is done to limit the nuclease activity in the cells. You'll get a burst of nuclease activity, and then it'll go away. So if you're transfecting mRNAs, just use the same protocol and same program that you're using for the transfection of DNA in your cells, and that'll work just fine. And that'll also allow you to transfect in the DNA template, but we'll have more discussion of that in a bit. This in vitro transcribed mRNA must be translated, so it has to be capped and polyadenylated. And the optimal amount, you may want to titrate that, but the range is from 0.4 to 4 micrograms of mRNA in a 20 microliter transfection. And you might need to use a significant dose. Many times there are endogenous RNases in the cell, so if you transfect in a small amount of mRNA, it may just get degraded before it gets translated. So you might need to transfect a larger amount of mRNA to compensate for this and overwhelm those endogenous RNases. And you might need to definitely optimize the ratio of nuclease mRNA and substrates. And also, Sleeping Beauty and Piggyback, these are the transposase-based systems. Those mRNAs have also been transfected very effectively using nucleofection. This is a slide about protein transfection with Cas9 RNP. This is from some of my friends at Sigma who gave me this slide. And basically, they're looking at... Oh, that's not me. I also have that alarm. I was thinking, what did I do? They were transfecting different amounts of the Cas9 protein here, and then different molar excesses. They 1 microgram versus 5 micrograms of Cas9, and then 5 molar excesses, 2.5 molar excesses versus 1 molar excess. They checked, they examined indel formation with the cell 1 assay, and then were able to quantify this. So in this case, you can see with 1 microgram of Cas9 and 5 molar excess, they got 32% indel formation. They dropped it down to 2.5, and it went up to 44. They dropped it down to 1, and it went up to 47. However, they got contradictory results when you look at the 5 microgram sample. And at the 5 molar excess, they got 63. Actually, the 5 and the 2.5 molar excess, they got 63, which implies that the system was saturated at this point. But then they dropped it down to 1 molar excess, and it dropped down to 52% indel formation. I don't really know how to interpret this, except that all I can say is this, the whole CRISPR Cas9 situation is complicated. We don't really know everything that's going on here. And so it just means the system is complicated, and that if you want to maximize your indel formation, you might need to do a lot of playing around with the ratios and the amounts of the Cas9 protein that you transfect. And actually, as you'll see in the next slides, that this is actually not very much Cas9 protein. People are using significantly larger amounts than this. So let's compare the Cas9 protein versus mRNA versus DNA. So again, the big advantage of using Cas9 mRNA or protein is that they're labile. You put them into the cell, you get a burst of nuclease activity, and then it'll go away. The growing consensus seems to be that the longer these nucleases are active in the cell, the more off-target effects you're likely to see. And this is often the case with DNA. With DNA, you're doing a transient transfection, so the DNA is going to be present in the cells for 3 to 5, maybe 7 days. And all this while, they're going to be cranking out Cas9 and guide RNAs. So these nucleases will have ample opportunity to scour the genome and find less than perfect binding sites, which they can then cut. These are the off-target effects. So by limiting this, the activity of the nucleases by using something labile, like the mRNA or the proteins, you tend to get more specific results and fewer off-target effects. So that's something to keep in mind. Nucleofection of mRNAs is very efficient. It's a single-strand molecule. It coils up in secondary and tertiary structure, and it transfects like a ball. So we get very good transfection efficiencies, typically in the 90 to 95% range. So the vast majority of your cells are going to be transfected with your Cas9 mRNA. Guide RNAs transfect in the 99% range because they're even smaller, so they get in very effectively. So if you use the mRNA system, the vast majority of your cells will be transfected effectively with your CRISPR Cas9 substrates and will have the opportunity to be edited. Doesn't mean they will be edited, but they'll have that opportunity. Same thing goes with the Cas9 protein. Unexpectedly, it also transfects at very high efficiencies. I've transfected protein before. We've transfected intact antibody molecules, 150 kilodaltons, and these seem to get in at about 20-25% transfection efficiency. I thought we'd see about the same with Cas9 because the Staph aureus Cas9, for example, is 120 kilodaltons. So I didn't expect a really great transfection efficiency. However, these do transfect well, so I think they must have some cell-penetrating peptide-like activities, kind of like what we see with TAT or Tenopedia or some of these other genes that proteins that get into the cell very effectively. So there's something special about the Cas9 protein, but that's fine. If it gets in very effectively, that just makes our job easier. Also, because the guide RNAs are typically complexed with the Cas9 protein before transfection, when they enter the cell, they are immediately active. So from a kinetic standpoint, they're active immediately. The mRNAs are active after this, and then the DNA lags farther behind on a kinetic standpoint. Then on the transfection side, don't be shy about the amount of protein that you use. So Doudna she wrote one of the early papers on RNP transfection. And here she nucleofected 100 picomoles of her Cas9 protein, about 16 micrograms in a cuvette. That's a fair amount of Cas9 protein. And that's what where she was able to get effective gene editing. So if we start looking at some of my customers and where they're going with this sort of thing, I have one customer who's using 15 micrograms, pretty much the same as Doudna used in her paper, of Cas9 combined with 20 micrograms of her guide RNA, and this is a 5-to-1 molar ratio. So in this slide, we're really going to be looking at the molar ratios of the Cas9 protein that gets used. We recently put out a white paper with Chandra Miller's group at Washington University in St. Louis, Missouri. Chandra was head of the genome editing and stem cell core, although she's moved on since. But anyway, for this paper, she was using the 20 microliter strip reactions and used 8 micrograms. But if we translate that into the cuvettes to kind of keep everything consistent here, that would be 40 micrograms of the RNP in her cuvette reaction. And she used her guide RNAs complexed to the RNP at a 2-to-1 molar ratio. They also did DNA transfections, but I'll skip over that. Then in the Nature paper that I've been talking about with Matt Porteous, he used 30 micrograms of RNP pre-complexed with his guide RNAs at a 2.5-to-1 molar ratio, guide RNA to RNP. So you see a trend here. People are really using a lot of the Cas9 protein, and they're using high molar ratios for this as well, from 5 to 2 to 2.5. So ranging in among those areas. And finally, I had another researcher that I worked with just recently where we transfected in the Cas9 mRNA. And they had outstanding results here. They ended up with an 85% editing efficiency in human T cells. And I think this is where we're going with the CRISPR Cas9 work, is really maximizing the editing efficiencies. And that's where I'm going with this slide here. So although this does differ depending on cell type, and that's a big part because this is all responsible to the DNA repair pathway of a particular cell type, so you will see variation. But we have customers who are getting editing efficiencies, non-homologous end joining editing efficiencies in the 80 to 90% range. That's unbelievable. I couldn't believe this sort of thing was happening, but I've seen this now with a number of customers. Because the Cas9 RNP and the mRNA both transfect so easily, what they're often doing is backing off on the strength of the programs, re-optimizing using lighter programs. They increase cell viability, but they also manage to increase editing efficiency dramatically. And I think that's a major trick. Now, there is a problem here, because if you want to do HDR, you need to transfect in a DNA template. And when they back off those programs to get these excellent editing efficiencies, these programs often are no longer effective at the transfection of DNA. So what people have been doing is to maximize your RNP or mRNA delivery and the editing efficiency, and then deliver the DNA template independently of nucleofection using adeno-associated virus. So this allows you to keep the high editing efficiencies and then introduce the DNA template separately, and then take advantage of those high editing efficiencies. And the assumption is that if you have a very high non-homologous end joining editing efficiency, that this will translate also into a high HDR integration efficiency. So by using AAV for the delivery of the DNA template, you can maintain the excellent editing efficiencies, yet the DNA template can also be effectively delivered. So this is a new idea that's coming out, and Matt Porteous did this in his paper, and I see many other people doing this. From a regulatory standpoint, AAV is also well known by the FDA. Efficiencies of our CAR-T work, which would make us much more effective if you could edit more cells more effectively. This would, I think, assist in the doses that you would be giving your patients, et cetera. A lot of downstream consequences to optimization like this. So just to finish things up, what does this mean for your research? Nucleofection allows you to use the most relevant cells in your work, cells that respond more appropriately to stimuli because they express the right receptors and the right pathways. And these results are then more likely to translate into in vivo situations. Nucleofection is superb with the transfection and modification of all these clinically relevant cell types, such as human T cells, CD34 plus cells, mesenchymal stem cells, iPSCs, and now with the LV, we can also do clinically relevant cell numbers as well. Nucleofection can be used in conjunction with all the genome editing technologies, zinc fingers, talents, CRISPRs, Sleeping Beauty, Piggyback, very effectively, very easily. And we're moving into the clinical arena, moving in the clinical direction by offering advanced documentation, IQ, OQ capabilities, our logware software, and can't forget our GMP-grade ex vivo media for T-cell expansion. So a lot of different ways that that LONZA can work with you in your CAR-T therapies. So thank you for your attention, and I'd be happy to take any questions at this time. Thanks.